Hello everyone. Good morning for some of you. Good afternoon for others of you. I'm very happy to see such an international crowd joining our town hall today to give you a brief glimpse of what Arise has been up to for the past 3 months and we'll be planning for the next 3 months. And more importantly, I hope you actually stick around after the presentations because we will use approximately 45 minutes to ask you, what do you think? So to really get your feedback. And this is extremely important because Arise is a research infrastructure which we are building for, as you can say, the research community. So for you and for other additional stakeholders. So I hope you enjoy the presentations, but especially, raise your voice. Let us know what you think. And just giving you a brief intro into Arise, as not many of you have joined all the meetings. So for some of you, it might be your first. So Arise is really all about biodiversity, and there are so many species out there. We have basically a lot of worlds to win here because not every species has been identified. There's some unknown species, and let alone is it easy to actually know exactly what's out there in the field. So here's a glimpse, for instance, of the more the European biodiversity. And if people ask me like, well, what is it? Well, here you go. It's basically every living thing out there, and each of those species have their own role to play. We have the pollinators, important for our food production, but also the decomposers and the soil organisms, very important for our food production. In the underground, making sure the nutrient cycles are actually in a closed loop. So every single species has their own function. And so therefore, it's also important to know exactly which species are out there so that we can understand actually what kind of ecosystem services are being provided and if they are in danger, if they are changing, under which conditions that they are important. But to date, there's a few techniques that we want to focus on with Arise to basically have a very fast and accessible species recognition. On the one hand, this is of course DNA. DNA barcoding has been in existence for a long time already. And now we're really trying to provide both the means for the technology on the one hand, but also the digital component on the other hand, to make this very accessible, make it cheap and easy to do barcoding, all the way from in the field sampling to the digital analysis afterwards, both on a single species as environmental DNA, which is of course also the future that we want to move into. On the other hand, Arise is focusing on other technology, sensors in the field. We have so many devices already. I mean, my phone can do a lot already. So why not use this also for consistent monitoring in the field? And so how can we actually use these sensors and create an infrastructure where you can plug and play using any type of sensor that can communicate with other data and digital services? What are these digital services? Well, plenty are, of course, around data management, as you will see later on in the presentations. Another component is also about AI services. So can we use artificial intelligence to help us out where a human no longer can because it's too much or too complicated? So, and how do we also make those AI services accessible by researchers so that even a computer scientist can go in, deep dive into big data, run huge pattern analysis, but also an ecologist can use state-of-the-art computer science models by click and play. So really make those two worlds come together. So that means that once all of this data is connected, we can really start making sense of our natural world, species, the species interaction, the ecosystem services, and basically the bigger picture. But that means that we are now building an infrastructure, both physical machines, digital machines, human beings interacted, and all of that needs to be interoperable. How do we do this? Well, we hope to not do it alone. Again, we are, of course, with a particular Arise team that is currently building the first foundational components, but this is also about you. This is about the research community also adding to Arise and other infrastructures also adding to Arise. We're organized in 5 teams. They're quite self-organized. They have their own assignments, but these assignments are interconnected. And you will hear more about this from the 5 team leads that will present today. Important to understand is that our approach is also to build end-to-end as much as possible based on use cases. So instead of just focusing on let's build the biggest database ever, we're actually taking one example in the field. So for instance, here you see the Diopsis Instax camera, which is one of our biggest examples. And can we actually build the components all the way from sampling in the field to the data analysis side? So that once we have this, we can copy it over maybe to other image recognition sensors or audio recognition sensors. And the same goes for the DNA side of things. So can we make a flow where we sample in the field, already start collecting metadata, have a solid identification mechanism, how to store the data, how to expose it and explore the data, and maybe communicate that data to other stakeholders. That's it in a nutshell. If you have more questions about Arise, do let me know, but I'll hand over gladly to the first team that's going to present what they've been up to.
Hi all, thanks Elaine for the introduction. So my name is Hanke Bakker, team lead from Team Sampling, but probably most of you know this already. So if we can go to the next slide. If we have a little of a recap of what Team Sampling has, its main goals, and in line with what Elaine just said about innovating all those techniques, it all starts with a reference database. We can build the best molecular recognition tools or whatsoever, but if you don't have a good reference, you're nowhere. So this is one of the core values that Team Sampling will add. And besides that, we also have a big picture of going from 5K to 100K samples per year to be processed. And this requires an end-to-end strategy for sampling. So this means from in the field all the way up to the lab until I can hand them over to Kevin for sequencing. But it also requires an end-to-end strategy for the storage, not the data, the images we take, but also and all the data we register, but also the physical specimens, because we always like to get back to the physical specimens and find out if we made a mistake or whatsoever. And I had a little picture here of all the processes, and I can tell you that it is a lot of time consuming. So we also will put some effort in reducing time. Because now we take like 15 minutes per specimen, and also keep the high throughput. So in the next slide, we see a little bit of an overview of our roadmap of where we are and what we did and where we want to get to. We focus on insect barcoding. You see target list on the top, but this is now in Kevin's team, so he will tell us more about this. We focus a lot on insect barcoding, so we have a lot of samples that we receive from specialists, courses that are ongoing, but also via other ways. We have Westerdijk also involved to do all the fungi barcoding, so this is a large ongoing part. And we also are testing in the field already, because we like to innovate these techniques, but we also need to know how efficiently they already are and how we can proceed on that aspect. So we're actually doing a pilot now in Amsterdamse waterleidingduinen. And besides that, there's a lot more going on, but that's just a quick view. So yeah, this is what happened in the last few months. We've been building our database, and it's actually live now. We can finally start filling our own database, a special segment in the collection database that we have in Naturalis. And here you can see some test records that are in there. And you can just scroll through everything and see all the data that is stored. And actually, you can see that there is a ton of data, very detailed, very specific. And everything is connected through this unique barcode that we use. Storage, everything is in there. DNA information can be stored in there. Gathering events, locations, whatever you can think about. So this will scroll slowly through it. It's maybe a little bit too long for this Townhall. But it gives you a little bit of a nice impression. If we can go to the next slide, maybe. Yeah, so this is also ongoing right now. We're in the field. Actually, I was there yesterday, just before the big storm happened, all the rain. So luckily I was out there quick enough. But we're in our second field work week, and the first week results are slowly dripping in. We registered 281 specimens in total. For the first field work week, 377 specimens are already photographed, and the first few hundreds are already subsampled, ready for Kevin. And we're also recruiting students now to analyze all the data. Hopefully, starting from September, we have 2 students. That's what we aim for. So it looks promising. In May, we also had the Myofauna course, which was very exciting. A a large group of specialists being here doing very specific research on those very tiny unknown species groups that are in the sand of the beach. Almost no one knows about them. The size is less than 0.5 millimeter mostly. But it was a great event, and actually with a lot of results. Over 530 specimens are subsampled, over 120 species, because they couldn't identify all the species to species level. So image recognition and DNA will help out here a lot. And we already have the preliminary information that we have one new species record, at least for the Netherlands, found in Katwijk. Those photos are this new species for the Netherlands. It's the 17th species of Gastrotriga, hairy bellies, fascinating. And we got probably 2 new species actually for science. So yeah, I'm looking forward to the DNA results actually. We're also looking into our processes. So a lot of time and logistic problems work is into getting specimens from field specialists and into our lab with correct data and whatsoever. So we're setting up a protocol for field specialists and trying to find out together with them how we can work more efficiently. For example, we go with numbers first instead of what collections at meteorologists used to, that numbers come last. So that's already a big change. And also we try to already put tissue samples directly in the buffer instead of ethanol, seeing how efficient this will be and how much less work this will be also for team sequencing. And also we try to define fixed templates to be filled in very efficiently. But after a short while, maybe we hope to go to a sampling tool. And this is also a work in progress. We had just a few workshop meetings, and this is a little bit of an example of how it will look like and work. And it looks very promising. So this is a little bit preliminary. They made a few nice artworks already to show you. Hopefully later this year, we can show you much more. I don't go through the whole process or whatsoever because it's just all baseline for now. So when we go to the outlook of 2022, we hope to have at the end of this year at least 10,000 species in total that are barcoded of the 43K that we know about. We hope to have the fieldwork completed and also the first data sets analyzed and documented. And we hope to have a lot of protocols for field specialists and to collect samples, but also the protocols for pilot studies like the AWD pilot. And also define and test the MVP for the sampling tool. So hopefully much more to talk about next time. So this is for team sampling.
Thanks, Marco. And let's hand over to the next phase. So Kevin, are you ready?
Yes, I am. Okay, well, team sequencing. So we're working on everything from DNA extraction to sequence data. So all the wet lab work and the bioinformatics. There's a sort of simplified drawing here of what the team entails. I'm not going too much details here, but you can ask questions about it later. Just to remind us of what our main goals are is, well, first of all, to create a DNA reference database. A centralized DNA reference database of that. And we've done a lot of projects over the last couple of years and data has been spread everywhere. So we're trying to organize that a bit. Also to create end-to-end protocols for every type of sample or specimen. And of course, we want to ramp up the speed, which we can process the samples as well as reduce the cost a lot. Which has been a bit challenging, but all the pieces of the puzzle are getting there, and we're trying to puzzle together the first set of protocols now. So it's probably on the next slide. Oh yeah, so this is our timeline. So we're working on a lot of different things. We have a very mixed team of people with different backgrounds. So we're working on barcoding, meta barcoding, but also on a lot of bioinformatics and data management stuff in the background. I will go into some of the details later. Yeah, so the next slide. So here's a quick update for the lab and the bioinformatics. We're working still on integrating some more advanced robotics in the lab, which of course have to be tendered because of European rules. But we have a manufacturer selected now, and we have the first meeting actually this week. So hopefully sooner rather than later, we need to make space in the lab to actually put these robots in, which is going to be fun. We've also been working a lot on the nanopore protocols. We sort of have a finalized version of the protocol now, all the way from DNA extraction to sequence analysis. There's been a few hiccups. We think the ghost of Sanger is haunting our lab, but we'll get there eventually. We had basically two big pilots running in the background as well, the marine barcoding and marine meta barcoding pilot. So we got our first specimens now for the myofauna reference, which Anko already mentioned. And for the soil meta barcoding pilot, we did an overseek run. So we have a huge mountain of data now. We just had a discussion this morning on how we are going to analyze this, because it's a big bone to chew on. And then, of course, we are working a lot on the target list, because we really need to make sure we know what we have to be able to know what we need to do. Which was an interesting challenge, because like I said before, the data is spread over multiple systems. We of course have the data that belongs to the specimens, so all the metadata. We have the sequences with some data attached to them, but we also have, of course, a list of species from the Dutch Species Register, which is not all super relevant, because there are some weird species in there as well. So we need to do a little bit of data filtering on all three of these data sets and sort of figure out how to incorporate them. So we now have all the larger DNA barcoding projects, like the FES barcoding from 10 years ago, and the WaterScan project from 5 years ago, and some other smaller projects. We also looked in the data that's already been published on BOLD, not just for the Netherlands, but also for Belgium and Germany. Yeah, just to have a bit of an indication of what's already been done across the border, just to see how we can sort of prioritize groups that haven't been done at all. And of course, we still need to incorporate the Westerijk data at one point as well. So we were figuring out how to sort of visualize this. Someone thought, well, maybe it's a good idea to collapse everything to family level, and then sort of make a nice graph that shows sort of a progress bar for each family. Then we realized there's a couple thousand families, so this chart is still a bit difficult to work with. So we went back to the basics. So behind this chart obviously is a table. So we now have our first interactive target list, which you can filter on all sorts of different taxonomic levels. But you can also sort the lists, and as you can see what's happening now, I don't know if it's readable. Maybe you need to increase the quality a little. Oh yeah, that's better. So we can sort the lists and basically find which are the largest groups without the least amount of coverage. So here you can see me switching between some different taxonomic levels. Of course, for species, the coverage is always 0 or 100 because there's only one species per species. And in this case, we're basically looking at the different families of insects, for example, to see which groups have the lowest coverage, which groups the highest coverage. But more importantly, which groups are the largest with the lowest amount of coverage, because that's the groups that we need to focus on first. Well, lucky for us, one of the groups that we are focusing on, or we were focusing on with some collection material, or the Ichneumonidae, a group of parasitoid wasps. And the fun thing here is, for instance, now, as you can see, we have all the those wasps selected, this family. and they either have a barcode or they don't have a barcode. So the next step is here. What we did is we can of course hide the ones that have a barcode. And so now we have a list of all the species that we are missing. And the fun thing about this sort of interactive table is there's a nice export table button there. And if you click that, you basically get an Excel sheet, which you can give to people that might be interested in collecting these specimens. It's basically how we can sort of build our target list custom to order. Yeah, and it's a pretty long list because it's a big family. So that's it for the target list. Of course, I can. It's just gonna show again now. We can of course show more later. It's not live yet. We're still improving a little. We're also still having some brainstorms as to what's the other kind of data we want to incorporate in this overview, right? Because now it's just an overview of all the species in the Netherlands, regardless of their status, and whether they have sequences or not. But we might also want to know if we have specimens or some other interesting data that we can link to it. For the outlook of 2020, 2022, so by the end, if everything goes according to plan, we will have integrated all the robotics into the workflow, and we can start ramping up the production speed. We're also working on the limbs in the background, which will hopefully have started sooner rather than later. And we want to have a first working prototype of the core sequence database. So the centralized database in which you store all the sequence data. And one of the more important outcomes of the pilot projects is that we want to have a protocol for high throughput soil sampling. And this is basically more about the sampling in the field itself, so that we can have a sort of a standardized setup in which we can analyze soil samples by knowing how many soil samples do we need to take per square kilometer or things like that. So that's where this big data set that just came in last week comes in. So hopefully by the end of the year, we can tell you all about that. And that's it for me.
Awesome. Thank you, Kevin. So Jacob, your turn.
Good afternoon, everyone. Nice to see so many of you. My name is Jacob Kamminga. I work at the University of Twente, and our team consists of members from University of Twente and Naturalis. The goal of DSI is to be able to identify any species from any type of digital media. This is an example image, which I think most of you will have seen. We want to support many use cases, so not just data types, so not just images, but also sound on the next slide. So it could look like this, where you have a spectrogram of an audio file. In this case, it's the same as a bird song. And the output of an algorithm will be the time the bird is heard and the frequency range. And of course, it's a species. So I just want to show you these different use cases. It's not just birds, but also fish, insects, and many different user scenarios. So some people may want to monitor insects, but other people may want to count wildlife, identify them from aerial footage. So how do we build an infrastructure that supports all of these use cases, right? So we're not focusing on one project, one algorithm, but we are making an infrastructure and tools to really speed up the development of all these algorithms. So we started with a repository. That's what we've built in the last year. And basically, this is where our story starts. So the repository allows you to find algorithms, but also if you're a developer, to help you develop your algorithm, to version it, to share it, or reuse someone else's algorithm. That's the core idea. For now, we are using it as a starting point to deploy algorithms. So we are currently focusing on automated deployment of algorithms. So if you're an ecologist and you don't know, if you don't have a technical background and you don't know how to use a compute platform in detail, we want to make it possible to click a button and it will deploy the algorithm for you. So we are currently focusing on an end-to-end workflow. That means we are going all the way from the camera to analysis output. In this case, you see a diopsis inside camera, images come in, an algorithm is automatically deployed, and results will come out. And the next slide, we are showing that simultaneously we are working on sounds, right? So we want to use the same tools, the same infrastructure to also be able to process sounds. So together with the BioCloud team, we have built a few scripts that can import data from the diopsis cameras. And so these are the images that we can currently import, but also metadata. So these cameras, they are placed in the field, and someone has to record where the camera is, how it is placed, etc. And this data is stored at the manufacturer, and we can currently synchronize with the manufacturer to pull in this kind of data. Once we have the data, we now have a data browser, which allows you to inspect the data and to filter it, and of course, query it, which means that you can search for data from a specific camera, a specific date range. And the idea is that you create a data set from that data. This is an example of a synchronization. We haven't really worked on the user interface yet, so all that you see is quite raw. But this is an example list of sensors that we can pull in currently, and where they were deployed and when they were deployed. This is a screenshot of our current version of the data browser. So you can select different cameras, look at the images, look at their details, and create a data set. And here we give the data set a name. And on the next slide, we have the analysis job runner. This is a process we are working on where we can actually use a data set, and we use an automatically deployed algorithm to run the analysis. Now, this is quite important. Currently, we do this on Amazon, and the prototype works. We have run analysis with 10,000 images. This has also happened in connection to iRods, which is hosted with Surf, if you know about Surf. We can go to the next slide. So we want to make it very easy. It's also for ecologists. So it's currently 2 drop-downs. You select your algorithm and you select your data set and you click Analyze, and the rest is automated. But of course, you need to know what happens, right? So you need to have some insights. So therefore, we have built an analysis job browser. So what you see here are example runs of analysis. The top one is an audio analysis run, and the other ones are the diopsis analysis runs. You can now click on more details to see what actually happened. So what you see here is the number of detections per day. So if you are using insect cameras in the field, you want to see if they were functioning properly. This is already a nice first insight. So if you expect a number of insect detections per day, but suddenly at a certain date they stop, right? You don't see any detections. Then either the camera was faulty, it may have fallen down, or it just broke, or maybe the algorithm has started malfunctioning around data with that timestamp. So this really helps you to work through the end-to-end workflow of camera systems. You can now look at the results. So this is an example of analysis job results. The number here is the number of insect detections in the images that the camera takes. And if you go to the next slide, very recently we have added the ability to show the detected bounding boxes around the insects. So we are slowly working towards the image I showed in the beginning, where you have the bounding boxes and also the species name and the confidence level of the algorithm. At the same time, we are working on standardization. So we want to host many algorithms that are developed by many different developers. And of course, not everyone uses the same language. So we have to somehow deal with this. And of course, one way to do this is standardization. So we're also looking at different standards. And for audio and images, we are working on incorporating Audubon Core and CamTrap DP. So this is work in progress. And we are planning to write wrappers around algorithms to be able to host a large variety of them. In summary, what we have done in the last 3 months, we can now actually use the OPSIS data by synchronizing with Faunabit, the manufacturer. We have started experimenting with importing large amount of images. So currently we're using 10K, but we'll go up to 10 million soon. We have added insights by the screens I just showed you, job progress. We can now batch images instead of doing them one by one. So there's already a bit of speed optimization. You can look at the jobs you have run. And yeah, like I said, the end-to-end workflow, we can look at the numbers of detection per day and look at results, which of course is very important. So what will we do in the rest of the year? We want to finish the end-to-end analysis of images and sound as a prototype to show that this is working. And of course, now we are focusing to the OPSIS because we have full access to it, but we also want to support other types of cameras, for example, wildlife cameras. Today, we had a kickoff meeting with the Snellius team at the Dutch supercomputer. So that is now just started. So we want to automatically deploy on the supercomputer. First of all, for inference, to analyze large amounts of data, and by the end of the year or early next year, also to help people deploy, train new algorithms. This year we'll also work on backend services to connect to external parties. So I mentioned wildlife cameras. We are talking with the Agouti platform, for example, to be able to list and serve our algorithms in the repository to Agouti. And we will refine the AI model repository, start working on user interfacing. The graphical user interface is currently almost not existent, so a lot of work to do there. And a very important functionality of the repository will be the ability to evaluate algorithms. So if you upload an algorithm to do a certain task, it's very helpful to you as a developer to compare or benchmark your algorithm to others. And of course, in the pipeline of algorithm development, a very important step is data annotation, which is a very big story, which we'll start this year as well.
Cool, thank you, Jakob. Lots of stuff happening on your end as well. Handing over to the monitoring demonstration sites team.
Yeah, thank you. I'm Daniel Kissling. I'm from the University of Amsterdam and the Institute for Biodiversity and Ecosystem Dynamics, and I'm leading the monitoring demonstration sites team. Line, please, next one. Yeah, so just to recap what our aims are for that particular team, we are establishing a couple of monitoring demonstration sites in the Netherlands in different habitats or ecosystems where we want to deploy different types of digital sensors. So it can range from wildlife cameras or insect cameras to sound devices and radars. The main aim is to collect the evidence of the occurrence of species, basically in an autonomous and automated way. We want to demonstrate the innovation potential with these monitoring devices, different devices in diverse environments. And then we also have the task to connect to a variety of users and stakeholders. So maybe you have you recap, or some of you recap. We started in the first year, we had a number of sites, a bit more than we are showing here right now. But in this year, we decided to concentrate our efforts on mainly 3 sites. On the one hand, Amsterdam City or green spaces in Amsterdam, and specifically we have one of them is Artis Royal Zoo, where we focus on urban biodiversity, wildlife, so urban wildlife within zoos and green parks. We focus on rat monitoring, for instance, and insect biodiversity, but also on monitoring bird and insect migration with radar. We have another site in the Amsterdamse Waterleidingduinen, where we have stakeholder Waternet. We focus there mostly on dune biodiversity and specifically on the role of mammalian grazers like rabbits and deer. There are exclosures that allow to assess, for instance, the difference between grazing and non-grazing or different levels of grazing and the effect on biodiversity. And we focus on insects there then as well. And the third site that we are consolidating is the Oostvaardersplassen, where we work together with Staatsbosbeheer. It's a grassland and wetland biodiversity site. And again, there is also an interesting role of herbivores like cattle, horses, deer, geese, and how they are feeding on the vegetation. There's also exclosures where we can measure grazing, non-grazing, and how that then affects other levels of biodiversity like insects. Briefly, what kind of sensors we have deployed in the first year in which we are operating right now. We have 4 types. The first one is wildlife cameras. We mostly focus on the Sniper Commander 4G wireless, which is a wildlife camera that works with an infrared sensor and an infrared flash. So that means if a warm-blooded animal like a ground-living mammal or bird is passing by, the sensor is triggered and it captures about 5 images in a row. And we can run these types of cameras in an autonomous way with solar panel and 4G data transfer so that the data are directly uploaded to a server. And we have tested what you see in the right corner, regular and wide lens in those cameras, which give us a different view of the landscapes and the animals that are passing through. On the top, you see we have at this point running 12 cameras in Amsterdam at Artis Zoo. We have removed a few of them because they were not so well located. We have in Amsterdam Waterleiding down a few and in Oostvaardersplassen. And the plan for this year is to focus on consolidating and expanding the sampling. So we develop at those different sites now more comprehensive sampling designs so that we can tackle specific questions, for instance, that center around the effect of grazing on dune or grassland ecosystems. The next one is our audio devices. In the first year, we have mostly deployed as a starting point these audio moths that you see in the left upper corner. These audio moths are relatively cheap, low cost acoustic devices, very small, like a few centimeters. They continuously record different types of frequencies like audible and ultrasonic. And they are very simple. They operate with batteries and micro SD cards, so no solar panels. There's also no data transmission. So this was basically a starting point also because we wanted, for instance, to pair them with wildlife cameras so that we can use both acoustics and images to monitor mammals. This year, no, sorry, go please back. So in the top you see, so we have at the moment 12 installed, well, 24 roughly installed in Amsterdam at 2 sites, Artis mostly, and then Flevopark, another park. And for this year, we are planning to expand that audio monitoring with more sophisticated device that you see in the right lower corner, these BAC devices, which are real-time ecoacoustic monitoring units that can be deployed autonomously in the field with solar panel and an automated data transfer. And we have ordered them and probably get them later this year. Next one. So the third type of sensor is the radar. We are operating at this point one of these Swiss bird scan radar in Amsterdam at Artis. This is a vertical looking radar that allows the real-time monitoring of migratory birds and insects. So not specifically species, but species groups. And the data are then processed on board already on the computer, and therefore it has to run with main power, and the data transmission is with a wired network. For this year, we are planning to, or we have actually just received the second radar, and that will be established at Kijkduin in Den Haag, which is a very interesting site for bird migration, where also a lot of other data, like on-the-ground data from observations, are collected so that we can compare and annotate the radar information. And we have also, together with this new radar, ordered a camera which allows us an vertical-looking optical camera with a zoom lens, which allows us then also to take images of migratory species and use this as annotation information. And then the fourth type of sensor we have deployed are these insect cameras you already saw in some of the other slides. We've mostly focused on the OPSIS insect cameras, which have this yellow screen that attracts insects, takes pictures every so second, day and night, operates with a solar panel and a battery, and also has this 4G data transfer option. We have at this point 10 cameras operating within Arise, and these are 2 at Artis, 4 at Amsterdamse Waterleiding Down, and 4 at Oostvaardersplassen. And the main plan for this year is to compare the OPSIS insect cameras with traditional monitoring. So we have the sampling team, for instance, in Amsterdamse Waterleiding Down, an ongoing effort to compare those 2, but also in Oostvaardersplassen, where they're also monitoring with Malay strips. And then we also testing now different ways of deployment, because we realized from the first year that while a lot of insects are attracted, for instance, flies and mosquitoes, several pollinators are not attracted if you have this as a horizontal screen, as a vertical screen. So we are now testing whether it may improve the capturing of pollinators if we put the screen in a horizontal way. Next one. Yeah, so as you can imagine, this results in large data volumes. So for the few sensors that we have deployed in the field in the last year, we had, for instance, for the Diopsis cameras, with those 10 cameras operating 2.5 months, basically 1 million images, roughly 700 gigabyte. For the wildlife cameras, we have been operating roughly a little bit less than 50. We had 700,000 images and 452 gigabyte. The audio files are much, much larger, so they are getting in the range of terabytes. And even the radar has much, much bigger data loads. We are currently not storing the raw data, but mostly the derived data, which over a year is also amounting to 0.5 terabyte. So with these data volumes, we are currently developing the data streams for data archiving. Next one, please. And this is basically developed in the sense that we can, from the sensors, either with the SD cards or the automated data transfer, collect the information, restructure the data through a download function, store associated metadata. Some of the metadata come with the sensor, with the images or the sound files, but we're also adding additional metadata forms collected with information by the users in the field, and add this information then in an archive upload. We have developed a pipeline for storing or archiving the data now on Surf Sara, and specifically on the data archive. This is a mass storage system on tape. So cold storage, which is relatively cheap, but also requires to organize the data in so-called tar files. And this is a whole pipeline that we have now been setting up within this data stream. And maybe the next one is just a very brief overview. I don't want to go into the very specific details, but how we're structuring these tar files. So we organize them with JSON files where the metadata are stored, where we can get information from the digital files, but also from the user, site information, location information. And then we organize on the right-hand side in the different colors. You see basically we organize or structure those files along download dates, location information, deployment, and then all the digital file information, so that we have this structured way, recording with metadata, so that we can extract whatever information we need. Next steps, we want to expand the monitoring demonstration. At the moment, we expanded within the sites in terms of consolidating the sampling designs, but we also want to expand with other sites and also other sensors, for instance, in the aquatic field. And then another bigger topic is the improvement of the automation, so the pipelines for data transmission, but also for more autonomous devices. We are looking into Raspberry Pi solutions, for instance, for the wildlife cameras, and the insect cameras, for instance, for the moth trap cameras. But also we look into improving the data streams from the sensor via a virtual machine that we are setting up at this point, and improving the link to the SURF data archive, but also then from the SURF data archive to the BioCloud. And that links to the next team, which is the presentation of the BioCloud team. Thank you.
Thanks, Daniel. I guess I'll take over and as the team lead of the last team. So my name is Chantal Huibers. I'm based at Naturalis in Leiden, but as many of the other Arise teams, we're a very team across multiple organizations. So the main goal of the BioCloud team is to build the underlying infrastructure for Arise and an interactive platform for access to data and services. As you've already seen in the previous presentations, not so fast, Elaine, we are collecting a lot of data within Arise. Some of that is already stored in some places, but it's important for us to make it interoperable as well as findable and accessible by others. So this is kind of our end goal. I guess that's always a good thing to start with, to put the dot on the horizon. So eventually we would like to have an Arise platform where you can access data and services. So on the right-hand side in this diagram in the blue are end user services that we want to provide. So this could just be search for data and explore the data, but also access the algorithm repository that Jakob was talking about, or even construct and train your own algorithms. On the left-hand side, you see a whole suite of other infrastructures that are already existing. And this is only really a handful of them, because there's many out there that we really would like to connect to and leave data sometimes in native infrastructures, or make sure that we can deploy services to other infrastructures. All of that is going to happen through backend facilities like API services, as you see there in the green on the bottom of the diagram. And one of the main things that we are still working on now is how to align all that data and how do we manage and store that data. Then we can go, yeah. So this is just a quick overview to remind you of all the different types of data that we are collecting both within Arise and that we are connecting to outside of Arise. So there's a suite of different data types for a species, so things that you can measure on a species, but with that also comes information around sensor and deployment information, what kind of algorithm or version of algorithm that you use, user information, information from the lab and the sampling team of how they collected and measured information. And as I already said, we want to make sure that all this information is fair, so findable, accessible, interoperable, and reusable. Yeah. So we've been working on a few prototypes, and we've been really thinking about how can we do this best. We have a suite of structured and semi-structured and unstructured data, and we want to harmonize that in some kind of way so that it can interact with each other, so within different domains within Arise, but also with the external world. And we have a suite of minimal system requirements around provenance, traceability. We want to have data objects identified by unique permanent identifiers. We also don't necessarily want to bring all the data into the Arise system, but really link to external systems as well. And making sure that we properly handle ownership and access control. Now, we've built a few prototypes, we tested them, they work quite well, but we've also done a little bit of digging in how do we do this big data management stuff. And I think we've really come to the conclusion from a conceptual point of view that what we're trying to build here is a data lakehouse, and there are particular technical solutions to do that, in which we will kind of look into over the next few weeks, months. So next slide, Elaine. There's a lot of options. I guess that's a good thing, right? Maybe, or maybe not so good. So you clearly can't see the detail in this image, but this is an overview of all the available applications when you look into big data management, machine learning, linking data, storing data, searching for data. One's already missing, I see. So, you know, there's probably even a few missing that we were looking into. So it is a bit of a challenge to not try to test everything, but really make sure that we pick the ones that we need based on the requirements of what we're trying to build. Next slide, Elaine. So as I said, we have been working on a few prototypes. One of them was really based on putting the data in a graph structure, the other one in a relational structure. Both use Postgres for indexing, so for storing the metadata, linked to iRods, which is hosted by Surf for the management of the storage of the actual payload. We've been doing this mostly for the insect camera use case that previous team leads also presented, to really try to do this on an end-to-end. So how do we get images from a camera all the way through the system into a report that tells you what kind of species you observed at which date where. So we've got working prototypes, but the question remains whether these are really as scalable, extendable, interoperable, maintainable as we need them to be. And they're not data lakehouse solutions yet. So this is something that we're looking into at the moment, on either expanding this or at least comparing this with solutions that are out there. Then our timeline. So far, we've worked a lot on the data management architecture. We have the privilege of a data architect joining the team last month, so that has really sped up our thinking around that. We've been testing iRods at Surf and testing the prototypes, and for the next half year in this year, we're really looking into setting up the data lakehouse for both the images as well as the sequence data, and having a minimum viable product for that, and making sure that we also address the requirements for access control for that. So most of the work that we've been doing is on the back end. We are also looking into how to build the species reference database, which we want to build on top of the already existing Dutch species register, so Nederlandse soorten.nl, for those who are familiar with that. But needs a bit of investigation on how we can put or connect genetic data with that. So once we have the core sequence database MVP, we really want to look at how to connect that so that we can build that species reference database on top of that. Front end wise, we haven't really started actual development, but we do a lot of designing, and actually we don't do that ourselves. We have some awesome students that use Arise as their final year project. Elaine, if you can move to the next slide. So one of the projects was done by Sjors. He's a student at the Hogeschool voor de Kunsten in Utrecht, and he's really given us some great ideas for the design of the species reference database, or the Nederlandse soortenregister, in the future. So that's really exciting, because it also makes a good talking point for us to really start thinking about the functionality that we want to provide, and what this should look like. And the final slide for us, we've got big dreams. Our goals for this year is so to have that data management system proof of concept of the lakehouse solution ready for the insect identification use case, as well as the sequencing use case. At least have a plan on how to connect the barcode data for the species reference database, and really use the designs that we have so far on thinking about how are we going to set that up. And have a proof of concept for the Arise platform. So second half of the year, we will focus on some prototypes for the UI UX, starting with the services that the DSI team has already developed. So I think that was it from me. I'll hand back to Elaine.
Awesome. Thanks so much for the team leads for presenting all of this in well, a bit over 7 minutes per team, but still, I think all of you have a nice overview now of what's been happening within Arise generally, but also the past 3 months, and what the outlook is for the rest of the year. So I really hope you enjoyed the presentation. And of course, also a big thank you of all of those technicians, developers, researchers that are actually closely involved to Arise to make this happen. But now let's turn it around. It's up to you if you're listening in. We're ready for feedback. So I'm going to open the breakout rooms, and you can join one of the Arise teams or the generic breakout room to, yeah, spill your guts. Let us know what you think. Give us feedback, give us input, whatever you want. So you can start choosing.